All right, we've been trucking along. If you've been quarantined or just getting a little bit behind, we have the old Chapter 5 Gim kit that needed to be done, Chapter 6 term quiz, super easy. The Ed puzzle on DNA and what DNA looks like, that's due Monday. But if you have all that done, then you are ready for the newest thing, Chapter 6 book questions. We're going to get most of it done before break, but this will end up in January, the last slide, the article. So we have book pages and a website and a PDF for an article here. But as always, you want to go find your slideshow and press the square arrow. Double tap so I can edit. Now I can either open this link to open the textbook pages or go back and split screen with Google Classroom. Down, and then the top red document are the book pages. So we end up starting off here. And nucleotides are the basic parts of DNA, and they come in 4 flavors: adenine, thymine, guanine, and cytosine. The last part, the third part, is a base. And that's the part that can change. Or the P and the S here stand for something that's the same in all nucleotides. So each nucleotide consists of 3 different types of material, and you keep reading to figure out what those are. So early on with DNA, they didn't know if it was important, but Chargaff was a guy who figured out that something always happened with DNA, and so you're going to find his rule. The picture of DNA is decently famous, and several scientists were involved, so figure out who took the picture, and then who used that picture to figure out the shape of DNA, because we couldn't see the DNA. And so getting a picture of it was insanely tricky. The video here is about how she did that. If we move on, the shape of DNA, there's 2 ways to describe the shape, kind of the mathematical fancy way and then another simpler way. So that's here. And then how long would it be if you stretched it out end to end? Ooh, there's my typo. There's an R there for no reason. And so that's over here. If you just took all your DNA and stretched it out end to end, you would find that it is ridiculously long. Now, one cell's DNA is about 2 meters, 6 feet long. That's not the answer, but if you have 100 trillion cells and one cell has 6 feet of DNA, that's going to be a lot. Incomplete dominance can be tricky because we now break Mendelian rules, and we have traits that can kind of blend. They're not really blending, but both are showing up. So the example is a snapdragon flower. And so we'll get onto that in slide 2, but if you notice, all the letters are capitalized. So with slide 2, you have an example that's not the same as the book. So here we have a red and a white flower, and they cross. Every offspring gets instructions for both red and white, and they come out pink. On the cellular level, some cells are red and some cells are white, but we can't see that, so we blend it with our eyes. What we use here is numbers, and all the letters are capitalized because nothing is recessive. So if all you have is R1, you are red. If all you have is R2, you are white. If you have one of each, you are pink. So before, we had 2 phenotypes, dominant and recessive, 2 things that a gene could look like, but 3 genotypes, 3 different pairs of letters. We still have 3 different pairs of letters that can happen, but now those 3 different pairs of letters look like 3 different things. So you're going to fill out the square. So for instance, this first square would be capital R1, and that would be a red flower. And this square would be capital R1 from above and capital R2 from beside, and that would be a pink flower. You're going to fill out the square and then figure out the percentages. Remember, each box represents a quarter or 25%. Then we're going to learn about more rules about DNA being broken here with the kitty cat. One gene can influence many traits. So you got to see which 2 traits are influenced by one mutation. The other way around, one trait like eye color can be influenced by something else. And then height can be influenced by something else. There's a video about that if you need it. If you have trouble with this fancy Punnett square or the idea of these tricky inheritances, watch this video. Slide 3, we keep trucking along about how DNA gets turned into stuff. So the size of groups of DNA that are red, it's kind of hinted at in the picture, but it actually tells you And the fancy name for that, a high school word, is a codon. But each group of 3 codes for a specific thing here. We have 3 parts. We have messenger RNA, this long thing, and you can read about it in this paragraph, the messenger molecule. Then we have the ribosome here in the picture and transfer molecules. So they're in the pictures, but they're also in the paragraph. So transfer molecule and messenger molecule are in here, but they're not in bold like ribosome. Now, when DNA reading goes wrong, that's called a mutation. And there's 3 different styles if you check the picture here. It's pretty simple. It's also in the paragraph, but the picture does a pretty good job. What happens, though, what are the consequences of mutations? You have to read down here. So not all mutations do what you think. Well, 1, they don't give you superpowers, but there's 3 very basic consequences of not fixing DNA, not repairing mutations. Then over here, selective breeding and genetic engineering. We got to skip around. We'll talk about pedigrees later. But selective breeding and genetic engineering are 2 ways that humans change living things, plants and animals generally, but also microbes, to be helpful to us. Now, this wasn't helpful. This was just for fun. They made tobacco glow in the dark with genetic engineering. The difference really is the amount of time it takes to make changes. So you'll be more specific in your reading here. Slide 4 is from an article. So that's this PDF or the website link. You can click the blue link. And the video is helpful. The video will get you most of the questions, but CRISPR-Cas9 is a bacterial trick for DNA, and it does something for bacteria that it doesn't do for us. But what does the CRISPR molecule do, and what does the Cas9 molecule do? There's 2 parts of it, and so you have to read down here. So CRISPR stands for clustered regularly interspaced short palindromic repeats, which is why we abbreviate it, because that's no fun. Then we have to talk about a good and a bad use of CRISPR. So CRISPR, to snip or not to snip, can be very good, but also can be used in ways that it shouldn't. And then what does CRISPR stand for? I just told you. The last one's an opinion. Do you think we should use CRISPR on people? Why or why not? So after you figure out what CRISPR can do, you might have opinions there. Now, if you go to my website, under Chapter 6, we have videos that I showed you, but back on the home page, there's another video about CRISPR in case you don't have an opinion about whether or not we should use it on humans. So that might help you there. Under current chapter in Chapter 6, leading into our project in January, where we'll learn about domesticated plants and animals, there's a whole playlist for cats and dogs, and then another playlist for domesticated plants and animals. So this one is just all about cats and dogs and how we've bred them to be the way they are and how they actually sometimes change us. As always, email if you have questions or talk to me in class.